Today I'm going to be talking to you again about the role of excipients in amorphous solid dispersion. So in this case, we've heard from a number of different collaborators. We started really on the academic side and moved into the analytical side. Today I'm going to speak about this from an excipient supplier standpoint, and then we will move next into the real industrial production of solid amorphous dispersions. So it's really a nice put together piece of information. So I'm going to start with the Solid Solutions background. Unfortunately, you've seen a lot of this content 3 or 4 times today, so we'll call this a post-lunch review, and I will very quickly glaze through a lot of this information. Then I'm going to talk very briefly about excipient selection and key characteristics. What characteristics of excipients are we looking for when we want to make a decision about what our formulation should have in it? Then I'll talk very briefly about screening technologies, screening methodologies, and a couple of quick case study examples of things that we've done here in Ludwigshafen in the labs, as well as our regional labs, and then a quick summary and conclusion. So again, I guarantee you've seen this slide 4 times today, as have I. Again, this is really the challenge, and quite frankly, we've been seeing this slide for a decade, if not 2 decades, at pharmaceutical conferences. So again, here are our poorly water-soluble drugs, 70+%, depending on how you want to categorize them, of these new chemical entities are poorly water-soluble. And again, painting with a very broad brush, we have bricks. So again, the fine sand of Copenhagen being thrown into the water. Does this actually dissolve? Of course not. So these highly crystalline, high melting point, and high log P compounds. Then you have these grease or grease ball type compounds, typically large, floppy molecules, amorphous in nature, much lower melting points, high log P. And throughout the course of these 2 days, I realized that today we're speaking primarily about solid amorphous dispersions and polymer-based systems. In the afternoon of today and also into tomorrow, we'll be talking about lipid-based systems, and depending on the type of molecule that you're looking at, you typically find one of these types of technologies can work quite well. So again, the BCS classification system, and really we're focusing, again, on the dissolution rate limited, solubilization rate limited compounds. So moving forward, principle of solid solutions is again quite simple. And I like the example of coffee. So when I just made my coffee, I put sugar in the coffee. Sugar has very little risk of recrystallizing out once it's dissolved into the coffee. So again, it's a very similar type system. You want to take your molecule, your drug, your sugar in this case, and dissolve it within a polymer matrix. So if we look at this as if it's a simple system, again, as a liquid system that you're dissolving something in that everybody's quite familiar with from a very young age, it becomes a very elegant technique for how we solubilize some of these very tricky drugs. And I believe that there are a number of really interesting and esoteric technologies out there to get poorly soluble drugs into solution. But really, what's important on this slide is this part here. It's tablets. We work in the pharmaceutical industry. There's almost an expectation by the consumers of the compounds that we are producing that they go into a form that is logical and accepted. And whether this is a tablet or a soft gelatin capsule or a hard gelatin capsule, this is really the preferred form to get the drugs into this. So again, starting with an insoluble drug and a hydrophilic matrix, you put the drug into this polymer, you make these solid solutions, and ultimately grind them, mill them, tablet them, and form forms that we're ultimately familiar with. So I'm not going to read this, however, maybe just the first sentence. So to obtain a single amorphous drug polymer phase, a certain degree of solid solubility, miscibility, and kinetic stabilization is required. So this is something very important and why the excipients play a fundamental role in making your decision on how to formulate these types of compounds. So in the end, you need to have a very strong interaction, as shown by some of our previous speakers, to be able to prove that your drug is going to stay there in solution, and whether you can have a true thermodynamically stable system is really the end goal here, because then you don't really have to worry about things from a stability point of view. So again, a similar slide as was prepared earlier, and if we look at the rate of dissolution, and here we have our standard crystalline API. As you make it smaller, and I spent a number of years working on nano-sizing technologies, micronization technologies, and you're always fighting against thermodynamics here. And the best example I have of this, if you were to take, again, your sugar cube, right? So a cube of sugar, let's say it's about 1 gram, and you mill this to the size that the crystals are about 1 millimeter in size. The amount of surface area that you end up with is about this much. It's about the same as a business card. However, if you take this sugar cube now and you grind it into particles that are roughly 10 nanometers in size, you now have about the surface area of a football field or a foosball field, depending on which country we're giving this presentation in, right? And now once you have the drug fully molecularly dispersed in the system, you have all of this tremendous amount of surface area for the exchange of the drug with the dissolution media. And again, tomorrow we'll talk a bit more about dissolution media. And this is characterized by this Nose-Whitney equation here. So again, your rate of dissolution is very proportionate to your surface area of the solid material. And so in this case, the drug that has been dissolved within this polymer matrix. So again, there were some very nice presentations earlier talking about some of the many, many phases that exist. But if we, again, paint with a broad brush and show our three core areas. So in this case, you have a glass suspension. You have drug that is basically encapsulated by the polymer, but not fully solubilized. And this will be crystalline here in this case, and amorphous here in this case. And again, you're fighting this driving force that this amorphous material wants to go crystalline again. It wants to form into these different clusters. And then again, your fully glassy solution here. There's a number of energy barriers that need to be overcome to fully solubilize the drug, and whether that's done through hot melt extrusion or spray drying or some of the other techniques. Again, once you get to this phase, if you have the proper amount of molecular interaction, you can really stabilize the system and prevent it from sliding right back down. So this is just a quick animation and quick summary of amorphous suspensions versus solid glassy solutions. So again, trying not to fight thermodynamics in this case. So in these cases, you might have some molecular you will have molecular dispersivity. The interaction forces between A and B. So again, the A, so here you have all of these dark blue structures. These are your interactions between the polymers and excipients that you have in the formulation. And the green here is your API. If your interaction of the API is ultimately stronger than the interaction between the API and the excipient, you will again have clustering over time. So looking at this, you can see over time, it might be kinetically stable, but then again, you're looking at this at different temperature conditions, different humidity conditions, and ultimately, you get API that will start to crystallize out. However, if you make a true solid glassy solution where the drug is fully dissolved in the system, you now have the case where your interaction with your drug is strong enough with your excipient that you prevent this from occurring. So again, this is really the desired phase here for producing a proper solid amorphous dispersion. Looking at the complexity of it, they tend to get quite complicated quite quickly. So if we take a very simple example, here's Colidone VA64, which you heard about earlier. This is our standard polymer for solid amorphous dispersions. Then you have your API. The overlap, you have a number of phases that can be quite complicated over time depending on whether these are fully solubilized, whether you now have amorphous phases, and whether you have crystalline phases. And once you have crystalline phases, they can be multiple polymorphs. And this is everybody's dread is seeing a couple of extra peaks on an X-ray diffractogram. Moving to something more complicated, in order to make these into systems that can go into a tablet, in many cases, you now have to add additional excipients, whether these are solubilizers or plasticizers, and this can further compound the complexity and further compound the number of phases that can exist possibly over the course of time. So you also have one that starts and then one at the end, and where this actually ends up going. So again, these are quite complex systems that can be quite challenging to characterize, as we saw earlier, and then quite complex to keep them in that phase over the course of the time that the tablets are going to sit on the shelf, in the pharmacy, or in various different people's homes in different regions of the world. And again, Ferdinand's going to talk about this a bit more related to crystallization inhibition, but again, we see the classic model here, and storage stability is something that is, of course, very, very challenging. I think in this case, there's a lot of solid amorphous dispersion work that's done either through hot melt extrusion or through spray drying that uses basically the TG of the matrix to kinetically stabilize some of these formulations. And this, again, is quite a gamble, because if it's not thermodynamically stable, you will ultimately, you're fighting against thermodynamics, which, at least from an engineering standpoint, is quite challenging to overcome, and it's a bit of a gamble. So when we start looking at the excipients themselves, there's a number of different things that we want to keep in mind, a number of different heuristics. So in this case, again, thinking about the model of sugar dissolving into a coffee, the excipients in this case are your solvent, right? So we look at this as our coffee. Having a solubilizing effect and also an adequate capacity for the drug. So it doesn't do us any good if the polymer in this case has the solubilization capacity for maybe 1 or 2%, unless it's a particularly potent drug, because then you're either taking 8 tablets a day, 12 tablets a day, or the tablets are quite large. Again, we want to have a strong molecular interaction with the API in order to do this. The thermoplastic behavior is more specific to hot melt extrusion type work. And then we want to have a wide range of processability. So again, from an excipient supplier standpoint, it's very, very important that these are functional and that you can use them with common technologies to be able to make solid amorphous dispersions. And there are a couple of heuristics here down at the bottom. So, and I like this slide quite a bit because it shows how the complexity can aggregate and also some of the key parameters that we have to think about from an excipient standpoint. So again, you're starting with your active and your polymer. And in some cases, this works very nicely. Just drug dissolved in Colidone VA64, and then you have you mill it and you turn it into tablets with a couple of binders, disintegrants, and coat it, and it works nicely. But the reality is, in many cases, this doesn't always work, and a number of other excipients are needed, either as processing aids, so in the case of a plasticizer, so to be able to make this through a hot melt extruder, for example, to minimize how much energy is actually going in that could ultimately be destructive to the API itself. And also we have additional solubilizers that might be needed. These might be needed in order to solubilize within the polymer matrix itself, or these might also have a functional effect later when you are dissolving the extrudate matrix, and they can interact with either bile salts or they can interact with the gut and have a number of different penetration enhancements. So ultimately, these solubilizers also play an important aspect when formulating these types of things. And all of these different things up here are listed. So we're thinking about glass transition temperatures, melt viscosities, solution capabilities, hygroscopicity. This is one that's, again, very important and was touched on earlier. Many times you're making these formulations, whether they're capsules or tablets, putting them in an ICH container at 40 degrees Celsius and 75% relative humidity. This is quite a challenge over the course of 6 months' time to keep some of these drugs in system. Because even if we make the formulations now, and we test them, we stick a probe in them, no matter how magic it is, ultimately, after that moisture comes in, you have something that changes. It's a non-equilibrium system. In many cases, we wish it wasn't, but this is often the case, that over time, the humidity gets sucked up into the system and can change the actual thermodynamics solubility limit of the API itself. So here's the strategy. We start with an API. We start with a chosen polymer, we start with an additive. And I'm going to talk very briefly about a couple of examples of film casting, which, while a not particularly difficult technique, can be quite elegant and quite simple in order to give some adequate predictions of APIs. So again, key parameters here: solubilization capacity, appearance, miscibility, and you'll know in a minute about what I'm referring to. Then we start making the decision on how we're going to actually produce this, whether it be through spray drying, hot melt extrusion. Let's take the example of hot melt extrusion. We now have to think about processability. What temperatures are we going to process at? Is the drug going to be stable under these conditions? Is it going to degrade as a function of some of the other forces that are going to go into the API formulation during compounding? And then finally, what do we do with it afterwards? Do we have to pelletize it? Do we have to put it inside a capsule? Do we have to mill it and mix it with a number of other components and turn it into a tablet? So once you get to the extrudate phase and the milled phase and you now have all of these products, does it stay this way? Is it going to be sticky? Are there any other things that need to be added? And then finally, we have our final oral dosage form. And then I think everyone here is quite familiar with the things that are needed in the final dosage form. But again, this is just a rough example of a decision tree looking at a solid amorphous dispersion. And here, I'm not going to go through point by point on this, but ultimately what we're looking at is hot melt extrusion, spray drying, 2 core techniques in the area of making solid amorphous dispersions, and some of the things that are really important. Sometimes this has to do with what types of capabilities are available at the pharmaceutical company themselves, which ones can be contracted out. And also, the solvents is really the big difference between these two technologies. Most of these poorly soluble drugs are not soluble in water, so you can't really spray dry them from a water-based system unless you can fully solubilize them in the surfactant system prior to doing spray drying. So that's really one of the core differences here, but ultimately there's a number of different ways that you can produce these things, but again, it's similar characteristics that we're looking at. So excipient screening, I really, really like this technique because it's surprisingly good at predicting miscibility. So again, you start with your drug, you start with your polymer excipient, you dissolve them within a solvent, typically water, acetone, ethanol, any of these types of systems. You cast them into a solvent film, and you can do this really easily. You can just put it on a glass slide as a drop. And providing your drying characteristics are good enough, you can very quickly see whether you form a nice glassy solution or if it recrystallizes. The comedy of this method is actually surprisingly robust in the sense that you can do X-ray diffraction, you can do DSC. These are sensitive to maybe 1% API recrystallization. You will see even micrograms of drug on a polarized light microscope under high magnification, drug starting to come out of solution. Then you can take these microscope slides, stick them in an ICH chamber, and see what effect humidity has on this system very quickly. So this is a really great tool that we use within our labs anytime we see a new API and try and figure out which polymers and which solubilizers would work under these circumstances. So again, there's a couple of pictures here on this preparation technique. You can now actually do it as a transdermal film as well. You can cast it in little Teflon things and stick it right on your skin. So solubilization techniques go into many different areas. And here's just an example of some results here. So here's carbamazepine on the top and phenofibrate on the bottom. Carbamazepine here. You don't see much difference with the addition of a solubilizer, but again, you're looking at this through polarized light microscopy, and over time, you can very quickly see, oh, here's your crystals. They start growing at about this 20% level. And again, phenofibrate a little bit higher, but you saw that with the addition of a solubilizer, this was completely eliminated. I have a small anecdote I want to share about phenofibrate. I spent a good portion of my academic career a long time ago turning phenofibrate into nanoparticles. And after several years of experience, we were able to get a sizable percentage of this into a liquid-type formulation. I come to BASF, I start working with Andreas, and we start doing hot melt extrusion-type technologies. And within about 2 weeks, we were able to get 20–25% dissolved in a polymer, basically negating years of my previous work. But it just gives you an example of how these techniques have really advanced, and we're not talking about these types of really crazy systems anymore. It's quite straightforward to do some of these testings and turn it into a viable product. So again, looking at the phenofibrate system, embarrassingly, I have to say, here you're looking at the extrudates that were actually produced, and then again looking at standard things like DSC, and also it's a little bit of an eye chart, but you can see the differences between the fresh and the aged crystal growth over time. These were left at 40–75 for a couple of months to see if there was any discernible crystallization growth over time. So talking again about excipient selection, BASF offers a number of different polymers, and they do have a wide variety of lipophilicity and hydrophilicity. Starting at the top here, we have our standard colidone-povidone type chemistry, and also things like Calicoat IR, Calicoat Protect. These tend to be very hydrophilic in nature. In the middle, we have our standard, I would say, really great polymers for doing solid amorphous dispersion type work. They tend to be somewhat amphiphilic. You have Colidone VA64, Soluplus, of course, which we heard about a bit earlier. And then moving forward, you can have things like Colidone SR, which is more related to sustained release formulations, and then Calicoat MAE. And the ability to work with these, so for example, Soluplus and VA64, if you've never done hot melt extrusion, if you've never done spray drying, you could do this in a week's time with these molecules. I would say some of the other molecules that are on here do require a bit more finesse and a bit more experience to be able to work with them. And you can do some very interesting pairings with these types of things. For example, we did some experimentation some time ago, blending SR and IR together to get modified release of the extrudates afterwards. So these are basically an example of all of the matrix formers that are within the BASF portfolio. And again, here are some key parameters when looking at this. So glass transition temperatures, melt viscosities, plasticizers. And you can very clearly see that as you improve the physical stability of the system, you tend to decrease the chemical stability of the system. So it's a very important interplay to try and make this into an ultimately processable but a stable formulation over time. Speaking of processability, BASF also offers a number of different plasticizers that can work for these types of systems. Typically, a plasticizer is required for anything that requires some energy input. So hot melt is a good example of that. Typically, things like our Calisolv PEG portfolio and our Cali4P grade. So these are the poloxamers. These products are all made in the Geismar, Louisiana plant, and these are all used to reduce basically the processing temperature. So you can see clearly here, these are the extrusion temperatures for Calidone VA64. By adding, in this case, poloxamer 188, you can drop that by 20, 25 degrees. Same thing with things like Calidone K12, K17, Calidone SR. You can very quickly drop that. It's quite interesting with the right blends of plasticizers. You can do this at almost room temperature, which is quite interesting. And then again, you see some of the other examples on here. Then again, talking about solubilizers, we have a very rich portfolio of solubilizers at BASF. And this is something that has a particularly profound importance to, from an excipient supplier standpoint. So you see some of these molecules, and Monica very nicely showed these molecular structures earlier. And what's really important to notice is the complexity of them. So each one of these lipid groups and each one of these PEG groups that gets attached here, it's not just a single molecule, it's a distribution. They have a very important quality by design aspect to them. There's a certain level of batch-to-batch variance, particularly if there's a lipidic base to it. So if these are based on fatty acids and fatty alcohols, they're naturally derived. So at somewhere upstream, they're coming from natural sources, and the blend of them can change over time. And that's really where we, as an excipient supplier, are looking to really improve the knowledge in these products. And I really like the slide that Monica showed earlier showing Cali4 RH40, because these are the types of questions that we want to be able to answer. And what happens if you take a drum of something like Cali4 RH40 and you liquefy it and then you freeze it? Is it still homogeneous? A lot of these solubilizers come as a semi-solid. And depending on where you are in the world and what season it happens to be, they could be a liquid, they could be a solid, they could be a blend. And there are a lot of challenges that can happen in terms of just testing them for release and then ultimately knowing how they affect the performance of as a solubilizer later on. So these are the types of questions that we as BASF are equipped to know about, because this is our expertise, being able to manufacture and work with these different excipients, whether they're polymers or solubilizers or lipids. This is really where we want to add the most value. So just a very quick highlight here, Calidone VA64. I know that we showed Soluplus a bit earlier, but again, this is an example of an extrudate. I like to call it spaghetti, but a nice clear spaghetti of Calidone VA64 with my favorite phenofibrate dissolved into it. But again, this is a really important polymer, not only from a functionality standpoint and from a capacity standpoint, but also from a pharmaceutical acceptance standpoint. I think the FDA IID limit of this is about 849 milligrams per dosage, so essentially an entire tablet. And this is really important from a formulator standpoint, because we do have to make sometimes these decisions get made in terms of what's acceptable and what's been used in the past. And this is a molecule that has a very, very wide range of applicability. And you see also on here wet granulation, dry binding, subcoating as a film former. It's a really unique molecule that has a tremendous amount of functionality and also a tremendous amount of impact in the pharma industry, and has been used in a number of different key formulations. So again, looking at excipient selection and the different analytical tools that are available. I would say that, and I'm not going to comment on AFM because the presentation earlier was so fantastic. Light microscopy, SEM, DSC, X-ray diffraction, these are sort of the standard types of techniques. I think that one thing that's often overlooked, particularly when looking at these types of systems, we can learn a tremendous amount from a lot of the really advanced techniques that are coming out. And this really helps us from a fundamental standpoint understand how the interactions work between a polymer and API, and also how they interact with different solubilizers, plasticizers, et cetera. And hopefully to be able to predict whether a formulation is going to remain stable or not. However, depending on where the formulation is being done, whether at a very large innovator drug company, a large generic, a small generic, a contract manufacturer, we don't all have access to these types of really, really unique and expensive pieces of equipment. So being able to do this type of prediction with tools that are available and standard within pharmaceutical labs is also something that's very important to us as an excipient supplier. It's to be able to show that using simple tools, we can also derive some very important conclusions based on the formulations. I shamelessly stole this slide from Monica, highlighting the solid dispersions and solid solutions catalog of products that BASF has. So again, the matrix formers, all of the different polymer matrices, depending on the aim and the goal of the final formulation. A number of different solubilizers that work in a tremendous amount of systems. It doesn't necessarily have to be a solid. We'll talk more tomorrow about lipid-based systems and liquid-based systems and how these form micelles in different phases. It's another really important and interesting area of solubilization. We also have a number of disintegrants, so once you turn this into a tablet-type formulation, and you can even do some of these techniques already with the disintegrants in the formulation. I didn't really touch on that much today in the interest of time, but this is also something very possible. And then, of course, the plasticizers and other additives that can go into these systems. And we do have quite a comprehensive portfolio that enables the ability of formulators to work with these different polymers and to solve, ultimately, the problems of poor solubility. And a summary and acknowledgement. Again, I want to thank Andreas Gritsky, who's really been a mentor to me since I started at BASF, even based in the United States. He's really taught me a lot of these techniques and a lot of these advantages, and really, unfortunately, crushed my dreams of working with nanosuspensions many years ago by showing me techniques that were more elegant and simpler and ultimately solved the problems better. So I really want to thank him. And some key conclusions again: bioavailability, the interdependence and maximization of solubility and dissolution rate. The amorphous solid dispersions, particularly if you get a system that is thermodynamically stable, is something that is ultimately the end goal. This is really where you want to be, because you don't want to fight against thermodynamics if you don't have to. And again, this is the preferred system, and we have quite a portfolio, and we're here to ultimately help you. And any of the questions that you have, whether they're here today, whether we'll answer them today, or we want to set up subsequent meetings to be able to discuss these types of things. This is just the tip of the iceberg in terms of some of the data that we've generated over time and the questions that we've had. We sit in a very unique place here at BASF that we get to work and talk to everybody, whether it's on the academic side with the very different customers and different needs, on the contract research side, on the generic side, on the innovator side. So there's a lot of little bits of information that we've collected over the years to be able to help solve some of these really challenging things.
BASF. We create chemistry.